A humongous genetic study in ME/CFS has just been released with over 15,000 ME/CFS participants. And so we should talk about this. Now, this is an open access preprint, and the link is in the video description so you can grab it for yourself and you can see what I'm looking at. Now, the central institution is the University of Edinburgh, Scotland, with collaborators in Dundee, Scotland, and then groups all over the world. There's other UK sites, Netherlands and Finland and Estonia and the United States. So a huge, huge undertaking. Now, as I mentioned, this is a preprint, which means it has not undergone peer review. And peer review is an essential part of the scientific process. And I'm always hesitant to discuss articles before they've gone through the peer review system. But I'm going to make an exception in this case, which I do occasionally. And in this case, because this is a large whole genome study and it is sorely needed in the ME/CFS field. And so I think it's worth talking about even at this early stage. So the idea behind the project is if we run whole genome analyses in ME/CFS participants, can we find genetic variants that are seen more in ME/CFS than in the general population? And so if we find genetic variants that we see more in ME/CFS, that would probably give us a clue as to what pathophysiological mechanisms are causing the disorder. So again, we're looking at genetic variants associated with ME/CFS, things we're not as likely to see in the overall general population. So what the groups did is they invited individuals with ME/CFS to participate. Now, those individuals had to meet either the Kansas consensus criteria or the U.S. Institute of Medicine criteria. Both of those require the presence of post-exertional malaise, and the participants had to have a clinician diagnosis of ME/CFS. Now, they sent saliva collection kits to the participants' homes, which is great. This allows the research team to include individuals who were most severely affected by ME/CFS, those people that were homebound, and even people that are bedbound. And that's very important because very few studies do that. And then they did the genotyping. They used the UK Biobank Axiom array, which is used in a lot of people. Now they got more than 15,000 people, but to simplify the analyses just for this paper, they only looked at people with European genetic ancestry. And in addition to that, the sample was strongly biased towards females, like 84.5%, and white at 96.5%. So obviously, as this type of research continues, these analyses should be done in other ethnic and racial populations. But anyway, for this paper, they ended up with 15,579 ME/CFS participants. And that is huge. And you always have to have a healthy control comparison group. Because they utilized biobanks for the control group, they got over 259,000 control participants, just massive. So this is a huge data set with lots of statistical power. And I'm not going to go into the programs. That's just too much for this, but they used a system called Regini to do the kind of genome-wide association analyses. And I'll talk a little bit about that as we go through it. But let's go to the results. I'm going to show you the main figure right now. And this is figure 2 on page 11, at least in the document I have. That's where it's at. Now, this figure shows the loci where gene variants associated with ME/CFS were identified. So places in the genetic code where there were variants that seemed to be, I won't say uniquely, but more associated with ME/CFS and not found in healthy individuals as often. Now, the research team did many other analyses. They did subgrouping like male, female. They did infectious versus non-infectious causes. They attempted a large replication. They even did some brain gene tissue analyses. Really cool stuff, but it would take almost 2 hours to cover everything. So I'm only going to talk about this main analysis. All right. So for each gene, I'll give you a little bit of background about what we're looking at. So for each gene, you have two alleles, one from your maternal parent, one from your paternal parent. So you can have a max of two variants of a gene, but there may be other variants possible in the population that you don't have. Now, some genes have very few variants, and some genes have a large number of possible variants. And then some of these variants have been associated with disease or dysfunction. And so we'll call those pathogenic or possibly pathogenic variants. All right, now this figure you're looking at, it doesn't tell us what variants were coming up associated with ME/CFS. This is just telling us the locations, and the words are the genes where there are some abnormalities in the ME/CFS genome found, or at least something that separated them from the healthy control group. So again, these are the variants that are seen in ME/CFS more than expected. Now also, I think I kind of alluded to this, so I just want to be really specific that these words that you see, these are the genes identified as the ones most likely associated with ME/CFS. But I do want to be clear that there are other genes can exist in these loci. And so there use some analytic techniques to identify which genes to focus on in this particular report. So as you can see, they identified 8 loci with variants associated with ME/CFS. I'm going to give you my interpretations of the relevance of these genes to ME/CFS. The authors have their own interpretations in the paper. Okay, so my first impression is these 8 loci are quite interesting. And I'm going to go through, let's start at the left, let's work our way to the right, and I'll tell you what I think a little bit about each one. So on the far left, we have RABGAP1L. And this is involved in intracellular vesicle trafficking, which is moving necessary components around the cell, within the cell. And pathogenic variants in this gene would significantly impair immune responses and also neuronal health. Now, obviously, when you're affecting the intracellular processes, this could also impact almost any system of the body. So this isn't something that would be specific to the symptoms of ME/CFS, but it is possible that dysregulation with this gene could cause symptoms of ME/CFS. So it's an interesting one. So next is BTN2A2. This one's more targeted, I guess. This gene is responsible for the regulation of T cell responses, particularly in suppressing T cell activation. And T cells are a major player in the adaptive immune system. And there's been some interesting research suggesting that T cells are not acting properly in ME/CFS. And so this genetic finding makes a lot of sense. All right, so next we have FBXL4. And this is critical in proper mitochondrial function. And mitochondria power basically all cellular processes. And if you have mutations, this could really impair a lot of cellular activity. But there's one thing in particular, that there's a specific mutation in this gene that can lead to encephalomyopathic mitochondrial DNA depletion syndrome, which severely affects the brain. I'm not suggesting that people with ME/CFS have that. I'm just saying that this gene is very important to proper brain function. And also, there are a few groups that are investigating mitochondrial dysfunction in ME/CFS. So the link with this gene is pretty obvious. Okay, so next one is SUDS3. I honestly don't know the clear link with this one. Now, the authors I read, they did cite one paper that shows this gene is important in reducing microglia inflammation, which of course is interesting to me and would make sense, but I'll have to read that paper and I'll have to get a little bit more background information. There's not a lot on this gene that I know of. Okay, next is OLFM4, and this is critical in innate immune response. So I talked about adaptive, and now I'm talking about innate immune response. And this is especially involving the neutrophils. And it also has complex roles with stem cells as well, and that gets really complicated. So if we talk about that, it'll be at another time, but very important gene. Then we have CCPG1, which is critical in autophagy, which is the cell's ability to recycle kind of the junk cellular bits. And research has suggested that this process is impaired in ME/CFS. So if recycling isn't working properly, you can get a buildup of free radicals that can cause inflammation and symptoms of ME/CFS. So that one is interesting. So then we have CA10, and this affects proteins that build synapses between neurons. So if you have improper protein building, that's going to affect neurological function. The authors note that this has also been associated with chronic pain. I wasn't aware of that. I'll have to look further into that as well. And I think there was a paper cited. I'll go back and see what that's about. But that's definitely interesting. And then the last one is ARF-GAF2. Interestingly, this again plays a role in intracellular vesicle trafficking, the endosomes. And dysfunction here has already been demonstrated to impair neuronal development in particular, so dysfunction in the brain. So I do see possible roles for this one, and I do see possible roles for almost all of these genes, but that's kind of the general story. So just noting some of the responsibilities of these genes, it's not really enough. It really gets down to the variants. But that's too complex for this introduction to the paper. And I think I'll wait for the peer review before I start diving into the variants, because it's possible that the analytic approach will change from reviewer comments. Now, also, I do have some questions about the study. For example, I'd like to know what percentage of ME/CF participants had at least one of the suspect variants. That would tell me kind of like how often do we see these genetic abnormalities in the ME/CFS population. So there are things I would like to see, but I'm going to hold off on talking about those in a video until the final paper is released as well. Right now, I just wanted to introduce this paper because it is a major milestone in properly large ME/CFS studies. So my interpretation, in my general view, what do I think? My first read of this is that this is identifying important ME/CFS subgroups. Some people, what it's looking like is that some people with ME/CFS may have an innate immune problem. Others may have an adaptive immune problem. Some may have a mitochondrial issue, and others may have a microglia issue. And those different causes, those may all fit in ME/CFS and that umbrella, but they may manifest with different symptoms, and they'll almost certainly need to be treated differently. So I think this is really important, and this paper identifies some intriguing places to look for pathophysiological mechanisms. But I do want to say that I don't think they will ever be a genetic signature for ME/CFS because there's too many possible first causes. So instead of there being the genetic cause, I don't think anyone believes that that's going to happen. I do think that in the future, genetics in ME/CFS will ultimately end up relying on personalized genomic analyses to kind of find these idiosyncratic causes or vulnerabilities to the disease. But these GWAS studies really helps us get started in identifying possible places to look more closely. Also, I'll tell you, I don't do much. I do a little bit of genetic stuff, but not much. So I'm more of a brain guy. But I'm really encouraged, and the reason I like reading these studies is I really like to see how the genes that are being identified fit with our observations in the brain. And I do really like the idea of the genetic people working on the kind of first part of the process. And then I'm working on the brain on the very far side of that pathological process. And we're working on both ends. And then hopefully we'll meet in the middle at the target that needs to be hit for new treatments. So I think all this work is important. I'm glad that there's people looking at this from all angles. So that's the main thing. By the way, this group I just wanted to mention in terms of just being really good general scientific community members, they also made the genetic information, the other data, and even the participants available for future analyses and studies by other groups, which is just great. I wish everybody would do that. So that's it. Just a quick introduction. Just wanted to share the news for those of you who hadn't heard that this really big paper has just become available. So congrats to the Edinburgh team and to all the other people involved in this massive project. And to all those watching, I will be back soon.